This morning, we're starting off with a wonderful lecture on evolution in the everyday world. Professor David Mendel is the Dean of Science and Harry W. Diana V. Henn Chair at the California Academy of Sciences. He is an evolutionary biologist with primary long-term interest in the molecular phylogenetics and evolution of the order and family of birds. His recent book, The Evolving World, Evolution in Everyday Life, won the 2007 Independent Publisher Gold Medal Award for the best book in the science category. And responding to him afterwards will be University Professor Rodney Honeycutt. Please give a Pepperdine welcome to David Mendel.
Good morning, everyone. Thank you, Chris. It's a pleasure to be here and join the distinguished group of speakers. And the title of the conference is Why Darwin Still Matters, and my talk is in that vein, but it's also really why evolution matters. And I wrote this book as someone that teaches evolutionary biology to science majors at the University of Michigan before I moved about a year and a half ago, and also to undergraduates that are non-science majors. And I was interested in trying to communicate to people why it is that evolutionary biology matters, regardless of whether there may be some segments of the population that have trouble accepting the facts that in many ways the scientific community has certainly moved on. It's part of our scientific background knowledge, and it is continuously tested, but it's also part of our everyday lives in ways that a lot of people aren't familiar with. And so what I wanted to do was just write about how we really are making use of evolutionary biology in what we're doing in a number of different fields. So that's what I'll talk about. And in doing this, I wanted to start. I'm going to try to move off the side here and see if you have trouble hearing me, let me know. I wanted to put evolutionary biology in a historical perspective of findings of science that have been unpopular. Certainly evolution has been unpopular. And I want to talk about three sort of case histories, and evolution will be the third one. The first would be the heliocentric theory. This was, when it first came up, was deeply unpopular. Here's a drawing of Nicholas Copernicus. I'm going to step out a little bit, hope you can still hear me, who published his work, De Revolutionibus, talking about how rather than the sun going around the Earth was orbiting the sun. This was published 2 weeks after he died in 1543. The work was actually finished almost 13 years earlier. He wasn't looking forward to this controversy of publishing this, and he was kind of worried about it. Even though it was a book for astronomers and mathematicians, he really put off the publication. The ideas had been circulating in the community of astronomers and mathematicians since about 1510. So it was held for quite a while. Written by 1530, the idea had been circulating since 1510, and it was published after he died, and there was a foreword by the publisher. Just the whole work was cast as just something that people who were doing some calculations for day length and things like that might be interested in. The sort of the significance of it as far as challenging the worldview of where the Earth stood within the solar system was really downplayed. Galileo Galilei really picked up on this and is remembered for having been the one that kind of suffered as far as pushing this unpopular theory. He published his book Dialogues in 1632, and shortly thereafter he was sentenced to prison by Inquisition. Even though, as many of you know, his daughter was in a convent, he did all he could to tell the church authorities that there couldn't be, if what he found was true, this couldn't be in some ways a contradiction to what the Bible was all about. But still, this was a tough one for church authorities to get over. Interestingly, Galileo's book Dialogues was on the index of prohibited books maintained by the Roman Catholic Church until 1835. In his book, of course, Galileo used this technique where you put your heresy in the mouths of other people. So you write it as if it's a little bit of fiction, but you're really getting your views out there, but you can sort of step back and say it's not really me, it's these characters that are saying it. But it was on this index of prohibited books for a long time. But we can say now that the verdict is in. And the Earth really does orbit the sun. And here's a diagram just showing how you can take a series of pictures this is based on Voyager 1, a satellite as it's traveling out through the solar system. And with a time series of photographs, you can map how the planets are orbiting. And it's really true that it's a heliocentric solar system here. And so if we think about how long it took this idea to pass from its first presentation in Copernicus's work until it finally came off the index of prohibited books, and we might say that's a very arbitrary, but it's a way of measuring when it was accepted by one of the most conservative audiences you might want to find. That's 325 years. Okay, so it's just interesting to think about it in this context. It took a long time for this unpopular idea to really become non-controversial within society at large. A second unpopular idea we might talk about is germ theory. And for the start of germ theory, which is linking human diseases to physical causes, not being due to divine retribution or divine smiling upon particular individuals, but that disease was due to particles of some sort, real physical causes. We can point to the work of Girolamo Fracastoro, who published in 1546 a book with the title On Contagion. And he really talked here about diseases being caused by what he called particles or germs. And he was noting that particular diseases would have outbreaks every year about the same time of the year. In particular kinds of habitats, you would find diseases. And so he was putting together the physical situation with disease, but he had no idea what the agent was. Nobody knew about single-celled organisms. They were too small to see with the naked eye. And this theory initially just went nowhere. He could see what was going on and had a lot of inference, but there was no real agent, so it wasn't convincing, even to the community of people that were trying to explain the natural world. The real evidence was provided by people like Robert Koch and his colleagues. Here's a picture of him along with some of the slides that he made for the anthrax spores. These are Bacillus anthracus endospores. Way over on the right. On the left in these slides are some of the bacteria themselves. And his real contribution was developing the stains so you could see these bacteria. And he very quickly was able to establish in people or animals that were sick the presence or absence of particular bacteria. This nailed the case for germ theory. It showed the direct link between particular bacteria and particular diseases. And you've heard of Koch's postulates when we're trying to determine the cause for a particular disease. You have to do these four particular steps, none of which it's hard to ever meet these Koch's four postulates in theory. Even HIV, it's hard to really nail these four down, but it's the steps you do when you're trying to establish that whenever you find the disease, you always find that particular species of bacteria or virus and none other. That's where it gets to be difficult. But this is the Koch behind Koch's postulates for linking particular organisms and the disease. So in this case, it would again be a little bit over 300 years if we're talking about the first presentation of this unpopular idea, which really rocked people's worldviews. It took about 335 years to accept, from the publication of Fracastoro to Robert Koch's work with colleagues, really showing the case that diseases were caused by physical entities. Okay, and evolution would be a third unpopular discovery of science, fact of science. And for the start of this, it was interesting when I was writing the book, trying to think about, who would I find as a very early proponent of descent with modification. And I found out a little bit about Benoit de Maillet, and I think he's a great person to talk about in this context. He was a French diplomat working in Egypt, so he made a lot of overland treks between the south of France and Egypt around the east coast of the Mediterranean. And he wrote a book with an extremely long title. Here's the title. Try getting a book published with a title like that these days. Teleomid, which is de Maillet backwards. So he's doing sort of the same thing that Galileo did by putting his heresy in the mouths of other people. So it's Teleomid, or discourses between an Indian philosopher and a French missionary on the diminution of the sea, the formation of the earth, the origin of men and animals, and other curious subjects relating to natural history and philosophy. He really outlined a theory of transformation over time of Earth and its inhabitants. And it's an amazing thing to read. It's clearly, he's trying to explain his observations of finding marine fossils up high on mountains while he's walking around the Mediterranean, areas that are currently xeric, very dry, high deserts. There's one passage I'll read you. Just recall, it was published after his death in 1738. Certainly it was written sometime before he died. And here's a short passage from his book. The transformation of a silkworm or a caterpillar into a butterfly would be 1,000 times more difficult to believe than that of a fish into birds if this metamorphosis did not occur daily before our own eyes. And the seeds of those same fish transported into marshes may have originated the first transmigration of the species and so on. He had no ideas about mechanism. It's laughable in hindsight, but he was trying. He was a naturalist. He was trying to explain what he saw, and he was postulating common descent in some ways, and that you could species were not immutable, and that one species could give rise to others. So he really did outline a theory of transformation, and his works were known to subsequent scholars. So here's an early evolutionary biologist, I would say. Of course, you will know who these two gentlemen are. I think Lamarck is really given a sort of a bad rap by history that is, by and large, undeserved. He was an evolutionary biologist. He published a phylogenetic hypothesis, which I'm showing right here. He's gotten many things right, including putting whales in with Mammalia, as opposed to Herman Melville, who kept talking about them as fish. And Lamarck is remembered for so-called Lamarckian inheritance, the inheritance of acquired characteristics. But that wasn't his innovation. It wasn't his theory. Everybody believed that at the time. Everyone in the museum where he worked in Paris believed in the inheritance of acquired characteristics. Darwin flirted with the idea and thought that this was quite possible in some cases as well. He changed in different editions of his work. So what Lamarck lacked was a compelling mechanism, obviously, and this was the contribution of Charles Darwin. But Lamarck got many things right, and he clearly had a theory for common descent for all organisms. And if you read Darwin was a wonderful gentleman, but for some reason he had kind of a blind spot about Lamarck. And he spoke disparagingly about Lamarck and his work and said it was terrible, he learned nothing. And that was in his earlier editions. It sort of softened up later. And Lamarck took a lot of heat. He was considered a fool by a lot of his contemporaries. And of course, he had famous battles with Cuvier, and so he lost to Cuvier, who was a much more successful debater than Lamarck was. But he really was an evolutionary biologist. And so, if we take the same approach, it's only been 288 years have passed since de Maillet's publication. And so when I'm talking to groups of teachers, I put it in this context to say, don't give up. It's still young. I think, science is on our side. We're going to prevail. For me, this is clear. It just takes a long time, and arguably, this unpopular discovery of common descent for all organisms is expected that it could take the longest time to reach broad acceptance because it's the most deeply personal. True, heliocentrism moved, the position of the Earth, but that's just our home, just our planet. It's not us ourselves. And germ theory has a lot to do with, causing diseases, but that's less about human origins. Evolution is all about human origins and our creation myths. And so it's going to take a while. But I think every step we take, every court case that gets won, every time somebody who's a high school teacher or a college teacher reaches out and talks about the fact of evolution and shows that it's not in direct conflict with a religious life or it doesn't make you an immoral person if you understand evolution to be a fact. We're doing the good work, I think, because evolution, it's just true. It's a truism. And the debate is not a scientific debate, it's about politics. So what I'd like to do is talk about some case histories of how evolution really is part of our everyday lives. I'll talk about a couple of applications of the evolutionary process. And in doing that, I'll talk about some what's called directed evolution. What I call directed evolution, it's also called artificial selection. I don't like the term artificial selection because there's nothing artificial about it. It's really directed evolution. You might talk about it as evolution in human hands. It's a case of coevolution. All evolution is coevolution in some ways. Organisms are responding to their environment and to other species. I'll talk a little bit about evolutionary computation, an exciting new field. And the second category of case histories will be applications of evolutionary history and how we use what we have learned about the history of evolutionary change over time for life on Earth in public health, meaning in understanding emergent diseases, how we use our understanding of evolutionary history to work towards a conservation of life on the planet and healthy environments, how we use evolutionary history knowledge to understand human culture, including, I'll talk a little bit about languages and about religion. And I'll talk about applications of evolutionary history in promoting justice through the legal system. And these are ways that people, even if they say, well, I don't accept evolution, well, you're benefiting by it. I mean, it's like if you don't accept science, you should turn off your TV and quit driving your car and don't get vaccinations when they become available, because it's already part of what is normal everyday life. Okay, so a little bit about evolution in human hands. And we have, of course, deeply influenced the evolution of all the domesticated plants and animals that we make use of. And so I'd say this is evolution in human hands, even though I'm not saying we knew what we were doing in terms of evolution as a process, but we had our species has had a very common sense approach to selecting particular organisms for those traits that we value, either in making domestic animals or crop plants, and even more than that. And so it's entirely reasonable to look at the gray wolf, shown down here sort of astride a map of the world, and see what humans have done as far as turning gray wolves over time into these different kinds of gray wolves, which we call dogs. As far as taxonomists go, it's all one species. There's no reproductive isolation between a gray wolf or a King Charles Spaniel, although I wouldn't want to be a female King Charles Spaniel trying to give birth to what may have been sired by a gray wolf or something larger. Say if you don't mix a female Chihuahua with a Saint Bernard, there'll probably be some troubles during parturition. But it is all, it's considered one species. And we can place the gray wolf within the context of other canids, as I'm doing in that one phylogenetic tree. We've got good information about when the first domestication events were beginning to start, about 12,000 years ago. There's some interesting discoveries from human burial sites where there's either pet cemeteries nearby or domestic dogs buried along with their owners. There's one interesting such archaeological dig that I was at outside of Tel Aviv. It's really a striking thing to see. It's the same kind of story with chickens. This is a species, there's actually more chickens on the planet than there are people. Earliest archaeological evidence for domestication of the chicken from the red jungle fowl over 7,000 years ago. The first evidence from 5200 BCE. And while the morphological changes are less striking in terms of size or something else than they are with the dog, the physiological changes have been amazing, because a red jungle fowl on average will lay 3 to 5 eggs, actually 4 to 6 eggs a year, but a highly selected laying hen will lay an egg about every day. So that's about 360 eggs a year or so. So that's a tremendous change just that people have done simply by allowing those individuals to contribute to the next generation who are laying on average more eggs per unit time. The same sort of story with Brassica oleracea, the cabbage. And I didn't really realize how variable and how much our species has gotten from developed the cabbage, which in the wild is a relatively small plant like this. It grows in the Mediterranean region. And in this diagram, I'm showing a series of vegetables that we have selected for and derived the cabbage plant from different parts. The turnip from the hypocotta, just at the top, the top base of the root. Rutabaga from the same general area. Mustard greens or kale from the leaves. The head cabbage from the terminal bud. Brussels sprouts from a set of buds. Broccoli and cauliflower. Also from a series of clusters of buds plus some of the stalk. White and purple kohlrabi. All of these things have been developed from the cabbage just through, this is evolution in human hands. People selecting for those individuals that have particularly enlarged features and promoting those in subsequent generations. And it really doesn't take long when the selection is very steep, very directed. So we've been making use of the simple fact that like begets like. That's all you need to know as far as a deep understanding of evolution over time. This is just inheritance. So this certainly has been a big part of the human story, enabling humans really to change and to develop culture. If it hadn't been for agriculture, allowing a little bit of food storing and more leisure time to do some other things besides simply subsist, our history would be quite different. It's the same process of evolution in human hands that we use to make attenuated vaccines. An attenuated vaccine is a vaccine that's just a little bit weaker than the wild strain. It's still the same pathogen, but it's been cycled, it's been forced to grow in an unnatural host species. What you're doing is you're forcing selection for some mutants that are capable of surviving in a new host species. That's a new environment. So you're selecting for certain mutations that allow it to live in a new environment. And when you take that organism and give it back to the original host, it's strong enough to survive and reproduce and elicit an immune system response, but it's no longer strong enough to kill you or make you sick. That's why the basic idea is here, you infect yourself to protect yourself. That's what a vaccine is all about. And so we attenuate it, we weaken it by growing it either in culture or a mouse or a rat or something like that. So we're selecting for mutants. Then when we give ourselves those mutants, they get our immune system going to elicit antibodies, but they don't kill us. That's the basic idea. That is evolution in human hands. That's been used in developing poliovirus, rubella virus, measles virus. And of course, there's been work on HIV viruses to try to develop vaccines, but those viruses are evolving so quickly that it just hasn't worked yet. And there's not as much work on trying to develop an HIV vaccine as there once was, but there are still 1 or 2 projects going on that. Directed evolution is a little bit different, but it's the same basic idea. It is a form of, well, certainly it's evolution in human hands. You can take a particular gene, and this is relatively new, a new area of human endeavor done in the lab. You take a particular gene that you know the function of. At the top here, this would be a linear gene, this line. You create a library of variants. You force mutations into certain sequences in multiple copies of this gene. You insert the gene into a vector, which goes into a bacteria, so you grow lots of copies of it. You screen the colonies carrying these multiple mutated copies of the gene for a particular property of interest in culture. You see how they do on a particular substrate. Do they have the traits for metabolizing certain substances that you're looking for? And initially you might see a very, very minor improvement or a minor change. But the point is you're going to do this hundreds of thousands of times this cycle. That's how evolution works, by very small steps. Some steps will be larger than others, certainly. But you iterate this in the lab, and you can eventually come up with something that you've really changed that particular gene. And then with genetic engineering that might be inserted in some ways to do things that we would like to do. You can enhance existing functions of particular genes, and here's some examples of how that approach with directed evolution is being applied. You can try to select for vaccine components that strengthen the immune response. If something's only a weak elicitor of immune response, you can select for a gene that's really going to have a big effect in eliciting antibodies. You can select for proteins that downregulate an autoimmune attack, and the whole group of these proteins are known as cytokines. Autoimmune attack is what underlies rheumatoid arthritis. People get this because your own immune system is attacking your own tissues. It's hyperactive. It's working, it's just working too well. Multiple sclerosis is an autoimmune disease. So if you could downregulate autoimmune attack by directly evolving better cytokines, this would be a good thing. And this is an active area of research. Inflammatory bowel disease. Everyone who suffers from inflammatory bowel disease, never mind. It's no fun. You can also select for enzymes conferring resistance to insect and fungal pathogens. A very promising field, and it's all about evolution. The more you know about the mutational process and where these mutations should go in different genes and how you can speed up the rate of mutations and what to look for when you're selecting those colonies that you want to put into the next generation of mutation and assessment, the better off you are. So the better we understand evolutionary processes, the better we'll do in terms of directed evolution on particular genes and proteins. Here's some case histories that have to do with our understanding of evolutionary history. Here's a picture of West Nile virus. And you remember when West Nile virus first showed up here in North America in 1999, people had no idea what it was. There was just, something was killing some birds in and around the Bronx Zoo. Some of the exotic birds the Bronx Zoo had were dying, as well as crows right around the Bronx. And there was no idea what happened. When you have a case like that, what are you going to do? You're going to call an evolutionary biologist. And you're going to isolate the pathogen, you're going to do some DNA sequencing, and you're going to plug it into a phylogenetic analysis. A tree of genealogy is shown here to figure out what it is. This is how we identify particular pathogens with an evolutionary approach. This is the best method for identifying what something is. And once we plug it in to a phylogenetic tree, here's West Nile virus at the top. It's a close relative of the yellow fever virus. Once you know that, you can say, oh, it's a flavivirus. Then we know that it's vectored by mosquitoes. Then you can tell, public health officials can tell people, hey, watch out for mosquito bites, take precautions. Wear long sleeve pants and long sleeve shirt, wear some bug bill, but put screens on your house. You can do something once you have this evolutionary information. Because then we've got a phylogenetic placement for it, we understand a little bit about its natural history. And it's been interesting to watch the evolution of West Nile virus as it has proceeded across the country. It arrived in California by 2002, and now it's down, I know it's down at least as far as central Argentina. And it has been, to a degree, ameliorated, becoming a little bit more benign. And this is predictable also, because it's trying to find an evolutionary stable strategy. If you kill your host right away, your progeny don't go anywhere. It's a similar situation with influenza A, in that an evolutionary approach to understanding what's going on with all the influenza viruses is very important. Influenza is a particularly challenging case because their genome is highly fluid. That is, you can think of it as coming in 8 cassettes, which you can mix and match in different organisms. And this is why you need a vaccine for influenza potentially every year, because you're seeing a different strain, a different kind of influenza. It's got a different, the genome is very, it's recombined at a deep level. And so if you really want to understand the evolutionary history, in this case, we're looking at particular genes, and different genes within a single virus particle will have a different history. And so an evolutionary approach helps us untangle this sort of complicated evolutionary history that influenza A has. And of course, when there's a new outbreak, one of the first things that's done is a sequencing study and a phylogenetic analysis to see what genes are involved in this new recombinant influenza virus. Evolutionary methods are among the very first that are used in looking at analyses for influenza A viruses whenever there's an unexpected outbreak in a particular area. So we're looking for new recombinants. I'm going to make sort of a quick point about what taxonomists are busy doing with all organisms on Earth is trying to develop a classification for them. And this is going to be deeply useful in public health and in medicine and understanding pathogens to the extent we can do that. What drives a classification? Well, now what's driving it is evolutionary history. We want phylogeny. We want the record of descent with modification in our classifications. Is that easy to do? No, it's not. Are there alternatives? Yes. And there's one that was dreamed up by the Argentinian writer, Jorge Luis Borges, which I really like. Because it's kind of fun. So there's a lot of ways we could organize organisms, what we see out there. This is one of the largest intellectual questions our species has ever faced. How do you make sense of all the life forms that are out there? What do you do with them? How do we know? What is going on? Is there any kind of pattern there? Well, here's one dreamed up by Borges. Those that belong to the emperor and bound ones, those that are trained, suckling pigs. I love number 6, fabulous ones. I have this disease. I think all life forms are fantastic. And number 14 is also quite nice. Those that from a long way off look like flies. So there are alternatives. We wouldn't have to do that. But as a species, we're figuring out that genealogy is a great way to organize things because we want to make use of that genealogical history, particularly in terms of public health and medicine. And, as I'll talk a little bit about, conservation biology. There are alternatives. Okay, a little bit about conservation. Here's pictures of 3 species that are endangered, that are protected: the humpback whale, the fin whale, Hector's dolphin. These are all cetaceans. You're not supposed to kill them, which means you're not supposed to eat them. But how do you know what you're getting when you go either to a food market or a fish market? Any guesses what it is in the top left? Tuna.
Tuna.
Yeah, okay. You can look at that and say, yeah, that's tuna. But what about when you're seeing something through a frost-covered cellophane package on top? Here's a picture from a fish market in Kyoto. You can read the kanji characters and believe what they are, or the English characters, but you're sort of taking somebody's word for it. How do you really know? Whether this, you can look at this and say, well, this is mammalian. But how do you know whether it's beef or whether it's a whale? You don't know, really. You're taking the word of whoever's labeling it when it's written. So here's a way in which evolutionary biology can help us in efforts to conserve endangered species. By going to a fish market, by basically doing some law enforcement. And this approach is being taken, and it's having an effect. You can go to restaurants, you can go to fish markets, you can go to butcher shops, you can take samples of what people are saying, this is a cow. Maybe it is, maybe it isn't. But there's a way now to track this down, to test for veracity in advertising with a phylogenetic approach. So I put this together as a sort of a composite from a couple studies. So if you had these mystery samples, the question mark sample from fish markets or food markets of some sort, that you're trying to figure out, are they really what they claim? Then you include that with some samples that you really do know what they are, and you see where these mystery samples fall out. And so in this case with the minke whale, these may or may not be legal, because the minke whale is protected in some parts of its geographic range, but it's not in others. And so what you really need here is some geographic markers that tell you whether minke whales are from, where their natal area is from. And there are people working on that. This can be done. The markers aren't quite there yet. It takes more work. This one is illegal. This is a humpback whale. This one's illegal. It's a fin whale. These are also illegal. This one turned out to be a horse. Turns out horse tastes it. So you can take the same approach when you're looking at caviar, which are basically fish eggs, which have, of course, DNA, and you can place them Some caviar from some species is legal, and from other species it's not. In my lab, we've been involved and got some funding from the Fish and Wildlife Service to try to identify geographic locales for gyrfalcons, because there is an illegal trade for falconry. And you want to know whether this particular gyrfalcon is from Greenland, Iceland, Yukon Territory, or someplace in Alaska, because it depends what the people's permits say where they got this bird from. And so this is possible. We've got a couple good markers. We need more, but we've got the beginnings of a set of markers to identify geographic natal locale for gyrfalcons. It's useful. We can finally put some teeth into these rules we've got for protecting endangered species. Because often all you see are the products from animals. You don't see the diagnostic traits of the organisms themselves. Another way in which evolutionary biology is useful for conservation, I'll talk about in this study also that I've been working on with colleagues and in my lab. This case of the, I don't know if you've heard much about the Asian vulture crisis, particularly about the Oriental white-backed vulture. This handsome, handsome beast shown right here. This species is going to go extinct in the wild within the next few years. And the population crash started in the mid-1990s. And prior to that time, this was one of the most abundant species of raptors, birds of prey in the world. It's a colonial breeding bird. There were millions of individuals in the populations, and suddenly they started dying off in large numbers, a dramatic decline. And to make a long story short, it turned out what was going on, the culprit was an environmental toxin known as diclofenac, which is the active ingredient in an anti-inflammatory drug that veterinarians prescribe for people to give to the family cow. It's basically Tylenol for livestock. And if you want to relieve suffering in the family work animal, in horses or cows, you can give them diclofenac, and it works for them. But when they die, and the range of the species is in Southeast Asia, particularly India and Pakistan. When the animal's carted off to a boneyard where the carcasses are taken for the vultures to clean up, the vultures cannot metabolize diclofenac successfully, and it causes what looks like gout. So you get uric acid crystals forming on the surface of the kidney, and it'll kill an Oriental white-backed vulture within about 3 days. There were a number of dead ends that were chased. People thought it was an infectious disease. It was a group in England that was convinced, but of course they never came up with the agent that was involved. We got involved in looking at changes in genetic diversity in the population as it was crashing, and comparing that with genetic diversity that we would find in museum samples, in what was left of the wild population, as sort of a benchmark for a captive breeding project. There's an individual that's not looking too good on the limb of the tree. There are 2 small captive breeding flocks. And we're helping to inform some of the captive breeding groups on which individuals to mix to maximize the genetic diversity in the captive flock. And so that's the way in which an evolutionary approach can be useful, because the way we map genetic diversity is using evolutionary approaches to looking at it, so we can help try to provide advice which will maximize the genetic diversity in the captive flock. And in this case, it's going to be hard, because individuals don't breed until they're 5 or 6 years old. They will only rear 1 or 2 chicks at most. Many individuals, they won't necessarily get along with each other, so you've got to find some pairs that are good pairs, or do artificial insemination, but there's at least a plan. In doing this work with Gyps bengalensis, is the Oriental white-backed vulture, we did a phylogenetic analysis for all the species in the genus, and what we found is basically a cryptic species, a species that was unrecognized, and so we can make arguments to give what had been called Gyps indicus tenuirostris, a subspecies. We can make arguments in a phylogenetic context that this lineage is as genetically distinctive as all good species are. We found the same level of differentiation between this lineage and good species. So we could argue this should be Gyps tenuirostris. And we and others have argued this, and it is recognized now as a slender-billed vulture, another handsome animal. And so this is recognized just in time to go on the endangered species list. But at least we can do something. These breed in remote areas, it's very hard to get into it. We've had a heck of a time trying to get the samples for genetic analysis because there's so many things, it's hard to get biological samples out of Pakistan, so it's the remaining colonies in Pakistan. One thing we found that works is if we tell them that we can't get any help from the Indian government. That really gets their juices flowing. Eager to help us. Say, well, we can, we'll step in, we'll help you guys. So here's a lineage which is distinctive. It would qualify certainly as a phylogenetic species, but also as a biological species. We can think based upon the inference that we have. So we can do something to try to preserve part of this biological diversity on the planet using an evolutionary approach. Evolutionary computation and algorithms. What these are basically doing is emulating processes of evolution to solve real-world problems in terms of design or engineering. So this is a little bit different. We're talking here about solving problems in the realm of engineering, but taking an evolutionary approach. So what do I mean by that? Or what are some applications first? And I'll tell you a little bit how it worked. And basically, some of you will figure this out. It works through mimicking the evolutionary process. But where has it been applied in particular? One example would be optimizing the orbits of groups of satellites. It's an interesting problem. Satellites need to be relatively close to the planet, say 100 miles off the surface. And in doing that, it's only seeing a fraction of the planet Earth. So if you're doing this for cell phones or for espionage, you want to see as much of the Earth as you can. So there's going to be multiple satellites, and you try to design their orbits for maximum coverage. How do you do that? How many, maybe you've got 20 different satellites you're working with. How do you maximize their coverage of the planet at different points in time to maximize this issue? That's a labor-intensive, difficult computational problem. It's also used in a lot of other communications issues. Optimizing scheduling for airline landings is another example. You've got departures to worry about, baggage routing, security clearances for airports, just the incoming and outgoing traffic. That's a real nightmare. There's just an example of what's going on in the US at a given point in time as far as airplanes coming and going. That's a computationally complex problem. Routing data and telecommunications networks, electronic voice recognition devices, stock picking, you can just stay away from that for a while. Predicting currency rates, playing chess, even scheduling classrooms during exam time. It's a computationally intensive problem. So there's lots of difficult problems to try to work on, and here's the basic steps that you would use in an evolutionary algorithm. It's similar to what I talked about in terms of directed evolution. You take a population of candidate solutions. They can be terrible, but you got to start somewhere. You just say, okay, here's some random potential solutions. You need to take those, set them aside, and look at which ones are relatively fit, are relatively successful, designate those as parents, subject them to mutational change. Those changed parents then you consider as the progeny. Those altered candidate solutions, the progeny, are a new population. You assess their performance, how they're doing, and if the optimality criteria, optimality is spelled suboptimally, if you've reached your optimality criteria for termination, you're done. Great, you can declare a victory. Probably you're not going to reach it very quickly, and you begin all over again. The power in this is in the iterations. You just let it go. And it's clear that an evolutionary approach to solving those difficult problems do much better, much more quickly than the other approaches that have been worked. It has been kind of a mini revolution in a lot of ways. Okay, so that's a little bit about design. There's a lot more. There's an entire field now of biomimicry where we're trying to pick up on features of organisms that have come about through evolutionary change that could be useful to humans. But we won't talk about those. I'll talk a little bit about now how evolution can be used to help us understand our own cultural backgrounds, to understand sort of human history. Here's a table which is taken from the work of linguist Morris Swadesh, published in 1952. This is a very early work looking at the relationships, the genealogical relationships for different languages. And this isn't biological evolution in the same way that we talk about when we think of descent modification. But it's looking at change over time in aspects of culture, and an evolutionary approach is helpful. That's the point I want to make. We're developing useful tools to help us know ourselves. So there's a series of languages here: English, French, German, Italian, and so on. And the translation, or the word for the same particular word, for three words: for the word "I" in all these different languages, for bird in all these different languages, and for fish in these different languages. You can see different degrees of similarity in those words. When you take these three words and turn it into 3,000, then you've got a sizable data set, and you can look for the relative similarity in this case, which doesn't, that's sort of a rough indicator of some historical relationship. Then you can begin to get an idea of the relative relationships for different languages. So this early analysis by Morris Swadesh, just to show how this field got its start. And more recently now, we're really getting some pretty sophisticated analyses using maximum likelihood statistics and large data sets for words to look at the relationships. This is still, we're looking here at similarity, and of course it's not neatly bifurcating. There's lots of horizontal transfer, there's lots of borrowing of words between different languages, but you're choosing particular cognates that you have some background knowledge, have not just been picked up. So it's increasingly refined, it's thoroughly evolutionary, it's developed by evolutionary biologists, and it's even gotten to the point where there are some very rough estimates for the time in thousands of years ago when these divergence events have happened. These are rough estimates. It's not my field. I wouldn't want to have to defend those. But they're moving into this area because there's an average rate that some people are beginning to suspect, try to talk about for change in words. To take this a little bit further, words go into books, and we can look at a book that has a well-known and very interesting history and see how it's changed over time. And so again, I think you can take an evolutionary approach just in looking at some of these attributes of human culture. So I put this slide together. This one is fun and gets me into lots of trouble. But I think you can take a work, and for me, Hebrew Scriptures, which I was bar mitzvahed and I learned a little bit about it. This is an account that people have written of deity. And I'm curious to take a look at how this has changed over time through a series of translations. And this is a highly selective chart. But there is a historical thread of change over time that runs through all of this. As Hebrew Scriptures have been translated into different languages, going into Greek with some new material added around between the years 100 and 200, these stories from an oral tradition were first beginning to be collected and redacted in the year about 1000 BCE. So it went into Greek, and then around the year 400 into Latin, in the Vulgate, the vulgar language at the time. Here's John Wycliffe's, the first English translation in 1384. And every time there was a lot of controversy about whether it was okay to do the translation or not. And at the time John Wycliffe was working, it was thought English wasn't a mature enough language. It was a vulgar language. It didn't deserve to have the Bible translated. John Wycliffe was teaching at Oxford when he started this project, and because he picked this particular project, he lost his job. So you have to be careful which projects you're going to use if you're trying to come up for tenure. He had to finish in Germany, and when he eventually got back, he was immediately condemned, and he was strangled just before being burned at the stake. So things didn't turn out too well for him. So you choose your study projects carefully. But every time, here's Martin Luther with the translation into German, there's going to be changes in the translation, which are kind of interesting. And it's just curious to think about these in the context of change over time and the consequences they have. And I'll just point to one example that may be known to many of you. in what's called the Old Testament or Hebrew Scriptures, there's a passage from Isaiah. The prophet Isaiah says to King Ahaz, "Behold, Ha'almah," that's the Hebrew, "shall conceive and bear a son." In the New Testament, in the Gospel of Matthew, this prophecy is restated, and in a translation, "Behold, a virgin shall be with child, and shall bring forth a son." There's an important difference here. In translation of the Hebrew Alma, ha is the article the. The Hebrew Alma to the Greek Parthenos, meaning virgin. Alma is actually translated as young woman, robust young woman in the prime of life. And there's no implication about virginity, as there is with Parthenos. So you might point to this as saying it's a mistranslation. It's a translation event that's had big changes in terms of the evolutionary history of particular groups within Christianity. And in a way, this is sort of like, might be a translational frame shift, or it's a problem in translation. We use that when we talk about molecular sequence information being changed over time and giving rise to new lineages of organisms. And this is just a very, rough parallel in terms of a metaphor. I think it's interesting. So we can take an evolutionary approach to help us understand some issues in the case of our own cultural history. The lost tribes of Israel are a really interesting topic. Of course, the 12 tribes of Israel were the 12 sons of Jacob. They gave rise to the 12 different tribes. 10 of them are talked about as the lost tribes. But really, when there was a civil war about 1,700 years ago, and 10 tribes went to the north, they were probably assimilated into the Assyrians. But there's this historical insertion point of the 10 lost tribes. What happened to them? There's been a series of groups. I mean, it's sort of a delicious opportunity for groups to say, hey, we're one of the lost tribes. And immediately you've got this deep history that you can claim for a relatively new group, if that's the case. There are a number of groups that have claimed to be descendants of one of the lost tribes. This includes the Nestorians of Mesopotamia, the B'nai Israel in Afghanistan, the Holy Shindai class of Japan, some Hindus, the Tutsis, and there were some Tutsis that were airlifted to Israel in the 90s, and it's still going on to a degree. The Lemba tribe of southern Africa, and in Mormon writings, probably familiar, talks about American Indians being one of the lost tribes. This is a literal gathering of Israel to be built upon the American continent. So here's some claims. What can evolutionary biology contribute to understanding whether these claims are true or not and helping uncover some of our cultural history as a species? Well, many of these claims are beyond testing, but it is possible to test some. And in the case of the Lemba from South Africa, some of the genetic evidence looking at Y chromosome linked traits, and the Y chromosome because within Jews, the priestly class, the Kohanim, breeds true. The Kohanim mate, marry, put it that way, other Kohanim. And so if, in case it has been Kohanim marrying within the priestly class, you would find high inheritance of particular Y chromosome, male chromosome linked traits. And basically, the oral tradition of the Lemba, that they are descended from sort of ethnically Jewish ancestry, holds up. There are a number of markers which support their claim. So in that case, it seems that there is something to it. So an evolutionary approach has helped us test some of these hypotheses about origins for particular groups of people. Some other ways in which an evolutionary approach helps us understand things. In this case, I'm going to move on and talk a little bit about the practice or trying to promote just decisions in court cases. An evolutionary approach is deeply integrated into all of this. And here's a little bit of background. Each individual shares much of its genotype with others, but it has unique identifying traits as well. We're all mutants, and those mutations are most likely in the most rapidly evolving portions of our genomes. Every one of you are different. And so we can look at those particular unique differences and call them a fingerprint of some kind, and there are combinations of traits that are polymorphic within populations, to identify populations of individuals, as well as new mutations within specific individuals. These are quite useful. These are well understood as a result of descent with modification that happens in every single generation. It's the mutation rate. And there are certain gene sequences that evolve quickly, we know as hypervariable regions. There's also others that evolve relatively slowly. It's good to know which is which, because we can make uses of them in court cases. The best use of these sorts of variable DNA sequences, which you may know as DNA fingerprinting, are in demonstrating somebody's innocence. Demonstrating proof is always a probabilistic thing, and the jury will ask you, are you absolutely 100% certain, beyond a shadow of a doubt? And you say, well, the confidence intervals on this are, 98.9. You mean they're not 100%? If a defense lawyer knows what he's doing or she's doing, they will ask you those questions, but often they don't. So it's best applied when somebody's innocent and they're just falsely accused. This is when it's very clear and there's no probability really involved. Since 1987, the Innocence Project, which was started by Barry Scheck, who was one of the lawyers involved in the O.J. Simpson case, he went on to redeem himself and do some actually scientifically valuable stuff. The O.J. case was a travesty as far as science goes, aside from everything else. Science never had a chance. It was all misrepresented, and it was just a matter of clouding it and devaluing it in the eyes of the jury, because it was a first case, 1990, or before that.
1994.
94, thank you. So I guess some of this was going on before the O.J. case. Let's say since 87. I got this figure of 245 yesterday off the webpage, day before yesterday. And so there's a lot of people on death row, or people who are serving life sentences, rather, who you can still go back to the evidence and find if the evidence that linked, if there's evidence that was used in their trial, or even subsequent new evidence that's come up to test whether they really are linked to the particular crime, you can look and see. And there have been 245 wrongful convictions for rape crimes have been overturned since 1987 based on DNA evidence. This has had a big impact just on the way in which some of these court cases are tried. Having post-conviction access to DNA evidence. A lot of times this had been blocked. It's getting easier. Expediting cases for wrongful conviction, it's now becoming more mainstream. You're going to do this. You don't have to argue so hard in each individual state to do it. There's tougher standards for eyewitnesses. We're realizing how faulty or how problematic eyewitness accounts are. Crime lab oversight. All the problems that came up in the O.J. trial. People are now documenting everything. There's much better understanding of how to do things in preparation for a court case. And the genetic profiling databases are growing, and an increasing number of rape crimes are resolved successfully. The limit of statute limitations on particular cases is being lengthened because you may want to go back. New evidence will present itself. The last case I'll talk about is an application of evolutionary biology within the justice system. And this is the state of Louisiana versus Richard J. Schmidt, an interesting case, a precedent-setting case, and one in which I actually had the opportunity and the privilege to be involved. This was the first ruling in a U.S. court on the disability of phylogenetic analysis. And so when I was asked if I would be interested in being part of the scientific group working on this case, I was eager to do so because of the historical significance, at least in one way of looking at it, involved in this case. So it was a dark and stormy night in southern Louisiana, in Lafayette Parish, in the mid-90s. And the uncontested facts in this case are that a gastroenterologist, 6'4", ex-Vietnam Green Beret person, went into the apartment at 2 in the morning of his former mistress and the phlebotomist that worked in his office. So he was a physician. Phlebotomist is someone who draws blood from your patients for you. And he gave her an intramuscular shot. He said it was a vitamin shot. She said it was an HIV cocktail. It had HIV. So there was the discrepancy in their stories. They agreed that he gave her a shot in the middle of the night. She went to the district attorney in Lafayette, Louisiana, and said that she'd seroconverted. She suspected that this guy, her boss, and that she had been having an affair with for 10 years. He was married, had injected her with HIV. The district attorney was a very astute guy. He got a search warrant right away. They went into the physician's office. They found in the back room a small vial of blood from one of his patients in one of those little tabletop refrigerators. You're not supposed to have those things laying around unlabeled. There was a light label on it, but it had not gone to the lab for the testing. He said it was for his own testing. This patient happened to be HIV positive of his. So they had an alleged source, a potential source for the HIV in the back room of this physician. They had all the viruses that are circulating in the victim, the former mistress, and all you then need to do, and once again, you're going to call an evolutionary biologist to figure this out. What happened? Whose claim was made? You take the alleged source, the viruses circulating in the victim, and you do a phylogenetic analysis together with an epidemiological sample, meaning a population sample of HIV sequences from other people with an HIV infection. And you see whether the vial of blood from the physician's office is completely distantly related at most to those circulating in the victim. If they're not closely related, he's innocent. Here's a diagram of just an HIV genome. We used evolutionarily one information about the relative rates of change. We used a relatively fast evolving gene in part of the envelope. The GP120 region of this envelope is evolving relatively quickly because it's in a race of sorts with the immune system. It needs to keep changing to avoid detection and elimination by the immune system, and a more slowly evolving region in the PAL, stands for polymerase gene. Just to show you the results quickly, here were 50 clones from the alleged source, viruses from the blood sample from the physician's patient. It has a sister relationship, a close relationship with those HIV clones from the victim. All the HIV gene sequences from, at the time, we just had 28, were further outside, and this sample's been increased a lot, and the same pattern holds. So this is interesting. This is the most quickly evolving gene, but you can see that there's an issue here in that, well, who infected whom? So if you're a defense lawyer, you say, well, how do I know that, the mistress didn't infect the patient, or something like that. There's nothing about direction of infection in that slide. Here's the gene sequence from the PAL region for reverse transcriptase gene. And there is a little bit of information here about direction of infection. If you put time on the horizontal axis, with long ago on the left and more recently on the right, the earliest divergences are found amongst HIVs from the alleged source, from the blood vial. Those are the older, the deeper splits within the tree. And so it suggests then that they're relatively new. These viruses are relatively young in the victim, and there is some information here about direction of infection. We told our attorney about this, and he said, wow, you guys are great. The jury's never going to get it. And so we had this information. We just needed to show the first slide, but we had this in reserve. There's a picture of Keith Stutes on the right, and here's the judge, Durwood Conk, on the left. The defendant was found to be guilty. There was other evidence as well. It's a pretty heavy thing to be testifying in a trial. It's going to put somebody away. So you really sweat. Have you got it right? Is there any problems? And of course, there were two labs involved, my lab at Michigan and the lab at Baylor College of Medicine in Texas. And when I got the stuff, picked it up at the airport, in between classes I was teaching, it was all blind label. I didn't know whose sequences I was working on. And it was similar at Baylor College of Medicine. So we took great precautions in doing all this work, and we didn't even know until the very end. We were doing the phylogenetic analyses before we knew how this was going to turn out. So these are the real heroes in the story, and it is a nice historical tie-in, and bringing full cycle the notion of how do the courts relate to evolutionary biology. So this is aside from the whole Dover case thing. I think this is looking at a pragmatic point. The courts initially had been used to keep evolution from being taught. The courts were thought, this would be the place to do it, to fight this cultural battle. And now, here's a case where admissibility hearings were allowing a core method of evolutionary biology, phylogenetic analysis, to be used in discerning, what happened, what's the actual sequence of events within a particular case. It's a powerful thing, this phylogenetic analysis. This brings up the basic point, is that science operates not on public opinion. It doesn't matter how people relate to a particular finding, whether it's popular or unpopular. And I talked initially about a couple other unpopular discoveries. Science works just by making discoveries, and this is when it's working at its best. It makes mistakes. It takes time to get those things worked out. And you continually test hypotheses to try to fine-tune them. But what we find is what we find, and we move on to the best explanations of the observations that we have. So I think, in closing, evolutionary biology is tremendously important, and it's deeply integrated into so many things that we do. And I've talked about how this informs our efforts at agriculture, and the better we understand evolutionary biology, we can do even better with our farming and working with other species. We need to start working with more species besides the very few domesticated species we've got. I talked a little bit about how evolution is involved in identifying and responding to emergent diseases and enforcing some conservation problems and understanding aspects of human cultural history, and then finally in figuring out some aspects involved in the courts. So thanks very much for your patience, and I'd be happy to try and take questions.
Before my general response to David's presentation, I want to thank him personally for coming, and I also want to thank all the speakers. This has been a really fantastic conference, and I want to thank the administration for supporting the conference, as well as our colleagues and students here. And I'm an evolutionary biologist too, so today Dr. Mendel has eloquently described the application of a theory first formulated by Charles Darwin. And Darwin's famous book refuted a metaphysical explanation for biological diversity by providing evidence that both supported evolution as such and destroyed the notion of the constancy of species. As shown by Dr. Mendel, the use of phylogenetics in forensics, medicine, ecology, and other disciplines in the life sciences is the direct result of Darwin's idea about descent with modification. Theoretical and empirical support of Darwin's second major postulate, mainly that natural selection provides a mechanism responsible for changes in populations, had to await the rediscovery of Mendel's laws of inheritance and the modern evolutionary synthesis. In particular, the modern evolutionary synthesis introduced the important idea that populations evolve as a result of changes in inheritable traits. As a result, population thinking, especially as it relates to changes in allele frequencies, has constrained the way we think about evolution at both the micro and macroevolutionary scale. Dr. Mendel has provided several examples of how population thinking has impacted many fields ranging from computer science to medicine to conservation biology. One obvious point from Dr. Mendel's presentation is that evolutionary theory has direct application, and both scientific and technological advancements have been made in the life sciences as a result of the theoretical framework provided by evolutionary theory. Does this mean that various ideas proposed by Darwin in the 19th century provide the last word on the process of evolution? Absolutely not. Many advances in the study of evolution have been made since Darwin, and ideas first debated by Darwin, Wallace, and others are still being addressed in considerably more detail today. Even many of the basic principles established by the founders of the modern synthesis remain somewhat unresolved and continue to create considerable debate. For instance, Darwin considered evolution to occur gradually over long periods of time, and this particular idea remained a component of the modern synthesis. Nevertheless, there are examples of rapid rates of evolution in genes, populations, and species. In terms of species formation, There are several examples of adaptive radiation, such as seen with Darwin's finches, that result in large numbers of species arising over short periods of evolutionary time. As stated by Dolph Schluter, founders of the modern synthesis formulated a theory of adaptive radiation, a rapid phenotypic divergence, as well as speciation that ultimately represented an outcome of divergent natural selection stemming from changes in the environment, resources, resource competition, etc. Not only do examples of adaptive radiation make the derivation of phylogenetic relationships challenging, they pose many questions that remain to be thoroughly addressed by evolutionary biologists. What factors contribute to the increased rate of species formation? What types of ecological and phenotypic differentiation accompany such bursts of speciation? How does one incorporate ecology into a consideration of rapid speciation, or for that matter, any type of speciation? What role does selection and competition play in adaptive radiation? As indicated by Dr. Mendel, molecular evolutionary biologists have used the concept of a molecular clock, which represents the accumulation of mutations in nucleotide sequences that coincides with geological time, and that's used to retrace the evolutionary history at both the macro and micro level, like the viruses, for instance. Nevertheless, not all genes evolve in a clock-like manner, and not all species or lineages experience the same rates of change. As a result of variation in the rate of change in nucleotide sequences, the focus of molecular evolutionary biologists has changed from the consideration of a universal molecular clock to more local molecular clocks. This begs the question, how much of this observed rate variation that we see is a result of adaptive shifts at the molecular level, and how much is simply the result of stochastic variation? Another continuing debate among evolutionary biologists is how to define a species. The biological species concept, introduced by one of my former colleagues, Ernst Mayr, and co-founder of the modern synthesis, is being challenged and even replaced or redefined today to try to incorporate different types of species that don't necessarily fit the original theory that he introduced. And they more or less encompass more of the evolutionary process. And even the lack of hybridization and exchange of genes between species, once considered important to the biological species concept, is being reconsidered. In some cases, hybridization may be adaptive, in that species can gain variation that might be advantageous in response to changing environments. In terms of species, what types of changes accompany speciation? Many early founders of the modern synthesis suggested that species formation should result in a genetic revolution, implying that species should show noticeable levels of genetic divergence. In more modern terms, a Nobel Prize-winning molecular biologist, Walter Gilbert, indicated that once the complete genome is sequenced, human genome that is, we will know what it is to be human. This appears not to be the case. For instance, a genome-wide comparison of human and chimpanzee reveals a divergence of 1.23%. This discovery prompted the authors of the genome sequence of the chimpanzees to ask the question, what makes us human? Clearly, the phenotypic differences between humans and chimpanzees are not the result of a genetic revolution and do not involve high levels of sequence divergence. In fact, my studies of local populations of pocket gophers from the western United States have revealed considerably higher levels of divergence than seen between humans and chimpanzees. Both Darwin and founders of the modern synthesis emphasize adaptation or selection acting on traits that enhance the overall fitness of an organism. Clearly, many phenotypic traits associated with an organism are adaptive and serve a specific function. Nevertheless, a large part of genetic variation within and between species is selectively neutral. Even recent genome-wide comparisons reveal that most changes in the genetic architecture of species are neutral to selection, with adaptive changes comprising, quote, only a small minority of the total genetic variation between species. Another issue that was reviewed yesterday relates to how much behavior is learned and how much it is hardwired by selection. One of my research interests has been the population genetics of the naked mole rat. From Texas, we call it naked mole rat. It's a hairless rodent living in East Africa that forms colonies that include a single reproductive female, a few reproductive males, and sometimes hundreds of non-reproductive workers. This particular social system is long-lived and presents a rather interesting dilemma. Unlike social insects, such as wasps and bees, the naked mole rat like termites, is a diploid bisexual species. Therefore, like Darwin, one must ask, how does such a complex social system evolve, especially if there are individuals that forego reproduction for the benefit of a few reproductive individuals? The evolution of such a system defies the ideas of competition among individuals and fitness as measured by the number of offspring produced during an individual's lifetime. This form of altruism is perplexing and cannot be explained simply by invoking either kin selection or reciprocal altruism. Like Darwin, evolutionary biologists continue to speculate on how such systems could arise under the constraints of current evolutionary theory. Also, like Darwin, we tend to waffle between explanations based on individual-based selection and group selection, the latter of which falls in and out of favor. Does the theory of evolution need to be extended to accommodate new findings from various disciplines in the life sciences? Absolutely. Darwin's entire theory was based on an attempt to explain the diversity of form in nature. The marriage of genetics with Darwin's theory changed our focus to a great extent towards genes and populations, and this may explain in part the rift between systematic biology and population biology, which are two general subdisciplines of evolutionary biology. Nevertheless, as evolutionary biologists, our ultimate goal is to explain biological diversification as it relates to adaptation and natural selection. By definition, this brings us back to the issue of form. Current research in evolutionary developmental biology, or Evo-Devo, I kind of like Evo-Devo, it kind of reminds me of Devo, you know, the old band. And both genomics and proteomics is rapidly changing our views of how complex forms originate. Rather than major modifications in structural genes, changes appear more subtle, in that they involve the timing of gene expression and or the alteration of interactions among various gene products. The question now is, how does one take information derived from this reductionist approach and reconstruct patterns of organismal diversity either within or between species? In addition, should such a reconstruction include a consideration of the interplay among genes, the environment, and organisms? Richard Lewontin, one of my former mentors when I was at Harvard, wrote a book called The Triple Helix, and I'll read you a quote that he made that really pertains to this. He said, "The trouble with the general scheme of explanation contained in the metaphor of development is that it is bad biology. If we had the complete DNA sequence of an organism and unlimited computational power, we could not compute the organism, because the organism does not compute itself from its genes. Any computer that did as poor a job of computation as an organism does from its genetic program would be immediately thrown in the trash, and its manufacturer would be sued by the purchaser." Of course, it is true that lions look different from lambs and chimps from humans because they have different genes, and a satisfactory explanation for the differences between lions, lambs, chimps, and us need not involve other causal factors. But if we want to know why two lambs are different from one another, a description of their genetic differences is insufficient, and for some of their characteristics may be irrelevant. Based on my current critique, one might ask, if there are so many issues associated with evolutionary theory that need to be resolved, what is the possible utility of such a theory? My answer is simple. Evolutionary theory is useful, as clearly demonstrated by Dr. Mendel. This theory, first proposed by Darwin, still provides the best theoretical framework for understanding the natural world. Given the complexity of nature, it is not surprising that we would continue to debate and to investigate issues that have not been completely resolved. As Dr. Durand mentioned yesterday, both the general theory of relativity and quantum theory are theoretical pillars of the physical sciences. Nevertheless, the two theories are not completely compatible. Does this mean that neither theory is useful? Absolutely not. Both theories have incredible explanatory power. Both are useful, and quantum mechanics in particular has contributed to considerable technological advancements. Dr. Mendel makes the same claim for evolutionary theory. It works, and it is flexible enough to accommodate current advancements being made in ecology, genetics, developmental biology, and molecular biology. Moreover, it has contributed to many technological advances that interface with nearly every facet of our lives. Thank you.